So remember, what we have to do is we have to take energy in the form of complex molecules, carbohydrates, proteins, fats, and so on. Okay. However, our cells cannot use that form of energy directly. It has to be converted into the form of energy, ATP, adenosine triphosphate, that our cells can use. And we should all remember that the organelle that can do this, and you remember what that is, of course, yes, it is the powerhouse of the cell. All right. However, do not use that term in my classroom. Uh, that is the mitochondria, and it will then convert ATP, convert all those complex macromolecules into ATP. And so the mitochondria are going to break down those complex molecules. That's a catabolic reaction. Breaking apart those bonds is going to release energy. We're going to use that released energy to make ATP via the electron transport chain and creating and maintaining a proton gradient. And then we're going to use that proton gradient to produce ATP as those protons diffuse down through ATP synthase, just like it did in photosynthesis. Okay? And again, um, one of the leftovers or one of the byproducts of this is going to be heat that we definitely do need. So there's our ATP, okay, and do look at that molecule. It consists of adenine, which we also find, which is also a nucleotide in DNA and RNA, and it's attached to a sugar, a 5-carbon sugar, and to 3 phosphates. Now, if there was only one phosphate on then that would be a nucleotide, the building blocks of nucleic acids. I do also want to point out that not only is there adenine triphosphate, there's guanine triphosphate, cytosine triphosphate, uracil triphosphate in RNA, and of course thymine triphosphate, which is found in DNA only. All right, and as you can see, it's that terminal or third phosphate bond that is a high energy bond, and when it is broken, it releases both heat and energy. Okay, when we break that bond. And remember, that's hydrolysis of AT, hydrolysis, using water to break apart the ATP. Okay, and the product of that is going to be an inorganic phosphate, and ADP, otherwise known as adenosine diphosphate. Okay, and you'll see it as adenine or adenosine. Either way is fine by me. I don't really care. There are several different ways to make ATP. We looked at using a proton gradient inside of the chloroplast, and we saw how ATP synthase used that proton gradient to make ATP. There's another way, and this is called substrate level phosphorylation. All phosphorylation means is adding a phosphate to it. And in this scenario, there's an enzyme plus a substrate molecule and ADP. And notice that the substrate molecule has an extra phosphate on there. So this is an enzyme catalyzed reaction. And the enzyme takes the substrate, which is PEP, which stands for phosphoenol pyruvate, and the ADP, and it takes the phosphate off the phosphoenol pyruvate, adds it to the ADP to make ATP. This is not a very efficient process, and our bodies use this to a small extent, but most of our ATP is going to be created using a process we call oxidative phosphorylation, and we'll get to that in a future lecture. So one of the key things that has to happen is that we have to have oxygen in our system in order to get the maximum amount of energy out of a glucose molecule. Now, we can still get energy out of a glucose molecule in the absence of oxygen. We're just not going to get all that much. We're going to be using that substrate level phosphorylation as we saw in the previous slide. only. We're not going to use oxidative phosphorylation, which occurs in the mitochondria. And as you can see, if there's no oxygen present, um, the mitochondria are not even involved. This will all take place in the cytoplasm of a cell, whether it's a eukaryote or a prokaryote. They all use this process that is called glycolysis, and we'll be talking about that. However, if there's oxygen present, then the pyruvate molecule will move into the mitochondria, and the mitochondria will extract as much ATP out as possible. Okay, these are two equations you must know. And if you look at them carefully, you will see that they are basically reverse of each other. We have already looked at photosynthesis, where we take two simpler molecules, CO2 and water, or carbon dioxide and water, and using the energy of light, we are going to make sugar molecules and have oxygen as a waste product. Okay, this is photosynthesis. Photo using light as an energy source, synthesis taking in this carbon dioxide from the atmosphere and converting that into an organic compound, i.e. glucose. Cellular respiration is the opposite. We take glucose and use oxygen to break that down and to make simpler compounds. So, once again, photosynthesis is an anabolic, energy using process. Okay, so that would be dehydration synthesis that produces sugars and heat as a byproduct, and oxygen, of course. Cellular respiration is a catabolic, exergonic, or heat or energy releasing process that takes the complex molecules and makes simpler ones. Alrighty, here's the big process. This is a diagram you should be able to draw. Okay, you need to know this one. And as you can see, there are three major steps to cellular respiration: glycolysis, the Krebs cycle, and the electron transport chain, which is also oxidative phosphorylation. So those are my three steps: glycolysis, Krebs cycle, and electron transport chain. Do notice that glycolysis and the Krebs cycles both use substrate level phosphorylation to make some ATP, whereas the electron transport chain uses oxidative phosphorylation. Okay, and as I said earlier, the substrate level phosphorylation, or SLP, only produces a small amount of ATP, whereas oxidative phosphorylation is going to produce a whole lot of ATP. And also do remember that that glycolysis occurs in the cytoplasm of the cell. It does not involve the mitochondria. It does not require oxygen. Okay? If oxygen is present, then the products of glycolysis, which is going to be pyruvate and NADH, will move into the mitochondria, where further extraction of energy will occur to make more ATP. If no oxygen is present, then the products of glycolysis, i.e. pyruvate, is going to undergo fermentation to allow glycolysis to continue. So let's look at the processes of glycolysis. Every single organism undergoes glycolysis. This is a very ancient pathway. This is probably the first energy producing pathway that ever evolved. Once again, it does not require oxygen. The early Earth, remember, did not have any free oxygen in the atmosphere, nor much free oxygen in the water. So we hypothesize then that along with other evidence, that glycolysis must have been the very first energy producing pathway. Why? Does not require oxygen. 2, it's found in every single living organism. 3, it does not require a complex cell or other types of organelles or anything of that nature. So glycolysis occurs in two major steps. The first one is the energy investment phase. And what we mean here is that we have to add energy in the form of ATP to glucose. So let's follow this. Look at the top diagram of glucose. Okay, we're going to phosphorylate glucose. That means to add a phosphate. What's the enzyme that is doing that? Hexokinase. Remember, kinases add phosphate molecules. And so we look at the second molecule in the white box, glucose 6-phosphate. You can see the phosphate attached to that number 6 glucose. Then we have another enzyme, phosphoglucoisomerase. An isomer, all an isomer has the same number of atoms in a molecule as another molecule. They are just arranged differently. And if you look at glucose 6-phosphate and fructose 6-phosphate, same number of carbons, same number of oxygens, same number of hydrogens, same number of phosphates. They're just rearranged in a different shape. And remember, in biology, shape determines function. So then we come to our second energy investment, and we're using the enzyme phosphofructokinase. There's that kinase once again, and it's adding a second phosphate to the fructose 6-phosphate. So now we have fructose 1,6-bisphosphate. 1 means that the numbers just mean the number of what number carbon it is, and the bi means 2. Okay, and that's going to make this molecule very unstable, and it's going to cause it to break in half, and it's going to make glyceraldehyde 3-phosphate and another one called dihydroxyacetone phosphate. But if you notice, there's another enzyme, isomerase. What's that do? Rearranges them back and forth, depending upon which one is in more abundance. If there's a whole bunch of glyceraldehyde 3-phosphate, otherwise known as G3P, then we're going to make some more of the dihydroxyacetone phosphate, and vice versa. All right, so that's the energy investment phase. Now we need the energy payoff phase. So here's the energy payoff phase. We are going to convert two G3Ps into 2 molecules of pyruvate. Okay, so let's start at the top, and we're taking those G3P molecules, okay, and the first thing we're doing is they're going to undergo a reaction using the enzyme triose phosphate dehydrogenase. What does that do? It's going to do a couple of things. It's going to add a couple of inorganic phosphates to the molecule, and it's also going to reduce NAD to NADH. What does that mean again? Remember, that means it's going to take electrons off of the G3P molecule and put them onto NADH. So NAD has been reduced to NADH, and these are high energy electrons. And NADH is going to be a product of glycolysis that's going to become very important a little bit later on down the road, and we will see that in the next lecture. All right, so now we have 1,3-bisphosphoglycerate, and we now have another enzyme coming in. Look at that, phosphoglycerokinase. Okay, kinase, once again, doing phosphates, but this time what it's doing, it's pulling a phosphate off the molecule 1,3-bisphosphoglycerate and adding it onto ADP. And that's right, guys, this is substrate level phosphorylation. And remember, there's 2 of these molecules, so this is happening twice, and so we're getting a total of 2 ATPs out of this. We now have phosphoglycerate, and we now have another enzyme comes in. It kind of rearranges it a little bit. And we're now at 2-phosphoglycerate, and another enzyme comes in, pulls off a water molecule, okay, rearranges the bonds a little bit. We now have phosphoenolpyruvate. That's that PEP molecule, okay, we saw in the other diagram or the other slide of looking at substrate level phosphorylation. And pyruvate kinase, the enzyme kinase, once again, is going to come in and pull that phosphate off, and it's going to produce 2 molecules of pyruvate and 2 more molecules of ATP. So what do we get at the end of glycolysis? Well, we're going to produce 2 NADHs, which will become important later, and we're going to produce a total of 4 ATPs. But remember, we had to invest 2 ATPs, so our total, or I should say our net gain of energy from this whole process of glycolysis is going to be 2 ATPs and 2 NADHs. And that's it, guys. I hope you had your notes and followed along. Take care.